Okay, welcome to video 2.5 where we discuss protein tertiary structure. This is a really important lecture and very fun, in which we'll basically be taking a tour of the highlights of protein three-dimensional structures, which of course form the basis of all of our understanding of biochemistry and molecular biology. Referring back to the previous lecture, here I'm showing how some of those super secondary structures that we discussed are represented in context. So here in this basic helix loop helix protein, what we see is the helix loop helix motif that I mentioned in the previous slide, and here we see another angle on that, and this is a little foreshadowing. I haven't mentioned before the idea of the leucine zipper, but in addition, one of the helices involved in that helix loop helix motif interacts with another helix through something called a leucine zipper. So here, in actual fact, we see one helix loop helix, and there's another partner helix loop helix here in gray, and what we see is that those two interact together, right, and they form a leucine zipper or hydrophobic interactions between the two helices. So anyway, the basic helix loop helix motif represented here, but actually we're forming a dimeric beta helix loop helix in which there are larger scale interactions, which we'll discuss later as quaternary interactions. Here is in human defensin 1 is a molecule which displays that beta hairpin, an anti-parallel hairpin, remember that? Another detail to point out in this structure, this is a very short little protein, and for these very small molecules we also see something called disulfide bonds that link the structure together, so we can hold together a structure that would otherwise not hang together through covalent means by forming disulfides between two cysteine thiols. Here's that that Greek key motif that I mentioned before, and I'll come back to that again. And here is triose phosphate isomerase showing us the beta alpha beta motif, in which we can assemble not the antiparallel beta strands that we saw here in human defensin 1, but parallel beta strands. Okay, so those are those super secondary structural motifs. Tertiary structures are assemblages of secondary structures and secondary structural motifs. This tour that I'm giving is broken into three sections really: alpha helical structures, beta sheet structures, and then combinations thereof. And then we might talk a little bit about some quaternary interactions after that. So I want to give you, this is really just an overview of these structural types so that you have a language to use when we're talking about protein structures as we proceed. First, alpha structures. These are helical bundles, and we've referred already to myoglobin, and we saw last time how we can look at the primary sequence of myoglobin, the amino acid sequence of myoglobin, and decide that that should be a helical protein, and in fact it is. When we look at the three-dimensional structure of the molecule, we see that it's comprised of all of these alpha helices. However, interestingly, in the case of myoglobin and in cytochrome B562 on the right, in a certain sense, the myoglobin serves as an armature for binding this thing called a prosthetic group. The prosthetic group of myoglobin is called a heme, and I've drawn a heme for you here. It doesn't matter exactly which heme this is for us. The big idea is that the protein itself, even though there are 20 amino acids available to it, is limited in terms of what kinds of chemistry it can perform, and so for certain kinds of chemistry we need to immobilize an ion, in this case iron 2, and we do that by presenting the iron to this tetraperol, this heme molecule. It's also referred to as a porphyrin, a very complicated organic aromatic structure, but itself only serving to hold that iron in place. And the iron can do a variety of things, including electron transfer chemistry, as well as, in the case of myoglobin, binding to and holding on to oxygen. So again, the idea is that in this case, at least, the protein seems to be a big extended structure for binding another kind of structure that then does the actual chemistry of the protein. So in any case, these are two alpha domain structures, or alpha helical structures. Here we have a couple of other kinds of alpha structures. Mu transposase, it doesn't matter exactly for us, for our purposes, what this is, but this is an enzyme that's involved in moving genetic material from one region of a chromosome to another. Here's GCN4, and I'm showing you this one. This is actually a very complicated structure. There's a lot happening. Let me back up and say that the secondary structural type would be the alpha helix. But in the case of GCN4, the tertiary structure doesn't really arise except in the presence of another protein. And so really we'd have to call this quaternary structure. That is a higher order structure than even tertiary structure. And it gets even more complicated. Not only do these two helices wind around together, but they also serve to bind a DNA molecule, which I've represented here. There's a lot of cool ideas here, but let's take them one at a time. So just in terms of the protein binding to DNA, we see the formation of both specific, that is, through the bases of the DNA ladder itself, and nonspecific through interactions with the sugar phosphate backbones. So specific and nonspecific interactions that allow the protein to bind directly to its ligand DNA. Okay, so there's a lot of nomenclature here, but it should make sense to you that a specific interaction with DNA would arise through that unique sequence of bases that define the sequence of the DNA, and nonspecific interactions would arise through the nonspecific repeating elements of the polymer, the sugars and the phosphates. We'll look more closely at that at the end of this lecture. The other piece of structure here that's really cool defines that quaternary structure, right? It's called a leucine zipper. And we'll see again later how there's a hydrophobic interdigitation of side chains from one helix across to the other helix. So there's a lot happening in this structure, but it's a beautiful alpha helical structure. We call it a coiled coil because it's actually forming a larger coil as the two protein molecules, the two helices, start to wind around one another. Okay, moving on to beta structures. Here's a protein that I love to show. This is green fluorescent protein derived from jellyfish. We won't go into that whole story right now. Nobel Prizes are involved, etc., etc. What I've done here in this so-called ribbon diagram is to cut away a small portion of the protein structure so that you can see the chromophore more clearly. The chromophore actually arises through the internal cyclization of the protein chain. So you can follow the trajectory of the chain here. It looks like we have a peptide bond here, and here's a tyrosine, but we have this glycine, tyrosine, threonine, and there's a cyclization step that happens, right? We form a bond between this alpha carbon and that alpha amino group of the peptide bond. What happens is we form a fairly extended pi structure, and we get a strongly absorbing molecule, and it happens to emit radiation in the green wavelengths. Now, the interesting thing about this that we'll explore later in the semester is the fact that we can tune the fluorescence wavelengths of this molecule just based on the sequence of amino acids involved in the chromophore and surrounding the chromophore as well. However, back to the idea of tertiary structure, this is a anti-parallel beta barrel protein, so we know that beta strands don't exist in isolation. They tend to wrap up into beta sheets, and in this case, the beta sheet itself wraps around on itself and forms a barrel structure, burying in the interior that chromophore. So it's very cool. You can see the chain winding through the core of that barrel and then coming back out again at the other end. So GFP beta barrel protein. I mentioned Greek keys before. Greek key referring to a motif seen in a lot of Greek pottery and other ancient Greek artwork, and we see this sort of key structure that winds around on itself and winds itself back out again and repeats again. And so in the Greek key structure here, this is from a molecule called gamma crystallin found in the lens of the mammalian eye. We see this sort of structure, and it's actually quite complicated. But what I've tried to do in the bottom here is color code according to the structure, and what I've done here. There's a couple of points to make here. First of all, these look clearly like mirror images of one another. We see repetition of the structure at larger scales, perhaps indication of gene duplication. This is an idea that I want to plant in your mind that we're going to return to when we talk about the evolution of biological molecules. So repetition in the structure, reuse of the tools in the toolkit. But now let's return to this idea of a complicated topology. So this is a bunch of anti-parallel beta strands. It's an anti-parallel beta sheet, but it's also a sort of a beta sandwich in which the sheet seems to fold up on itself. And by color coding the individual strands and mapping them onto this much simpler simplification here, you can see how the chain winds around on itself. So the anti-parallel strand isn't as simple as a bunch of hairpins linking it together, right? We see, although we do see a hairpin here, but the end of that chain is way back here, right? And then we wind way back around to come back to a green strand. And so anyway, a very complicated topology in gamma crystalline. And maybe rather far away, you might think, from the Greek key that we see in this Greek artwork. Okay, there's also some strange ones that are worth pointing out to you. This is the so-called pentapeptide repeat protein. You get the sense that the primary sequence of the protein is highly repetitive, and it gives rise to this parallel beta barrel, which is a very interesting structure and fairly not terribly unique, but we'd say rather unusual. Now, let's look at alpha-beta proteins. Here is the TIM barrel. I've mentioned it previously. TIM stands for triose phosphate isomerase. You'll come back to triose phosphate isomerases in biochemistry 2 when you talk about carbohydrate metabolism. But for now, it's an archetypal barrel protein. We see that it is a parallel beta barrel. How do we accomplish a parallel barrel? Again, it's a much more complicated topology in which the individual strands of the barrel are linked through an alpha helix. So there's a beta alpha beta structure there. I've tried to highlight that for you. And here again too, you can see the repetition of that beta alpha beta barrel as we wind ourselves around, allowing us to construct a much larger and more complicated molecule. A couple of other alpha-beta proteins that are worth mentioning. This is a fairly common structure represented here by phosphoglycerate mutase. Sort of a sandwich, if you will, of beta strand with the bread of the sandwich being these alpha helices. I call it these open sheets because they're not wrapping around to close themselves off into a barrel. And here ferredoxin is another, so maybe open-faced beta sandwich. There's helices on one side, the beta strands on the other. This one is really worth mentioning again because of a prosthetic group. Here an iron-sulfur cluster. There are other more complicated iron-sulfur clusters that we will consider later, but this is a redox center. Again, the protein serving as an armature for holding the prosthetic group in place so that the chemistry can be formed with non-protein atoms. Now here's a fibrous protein, collagen. I mentioned collagen in the previous lecture when discussing Ramachandran. This is his triple helix structure, which was borne out, the model borne out by fact or observation of the real three-dimensional structure of the molecule. You can see that it's helical in the sense that over a long term there's a repetition or a helical structure here, but really this is a more extended, almost beta-like structure. And we can see that there's interesting hydrogen bonding that's going on between strands, so interstrand hydrogen bonding. I've tried to represent that for you here. We also see that this structure is heavily glycine and proline rich, and we know already that proline is going to restrict our phi rotation. So we can get, this is part of how we can achieve this extended but highly interdigitated triple helical structure. Not soluble protein, right? This is a fibrous protein. Okay, now let's step outside of our standard soluble proteins that I've been discussing a little bit further and look at integral membrane proteins. I mentioned these in one of the first lectures. This happens to be the structure of rhodopsin. Well, this is a complicated picture, so let me describe what we're seeing. This is a mostly helical protein. We have some beta turns that tie helices together. The way I've color-coded this, however, is that nitrogens are blue, oxygens are red, and any hydrophobic or nonpolar atoms are colored yellow. So what do we see? Well, with rhodopsin we see pretty polar end of the molecule over here, pretty polar end of the molecule here, and a big expanse of nonpolar surface area. This is how we can tell that this is a nonpolar molecule. An integral membrane molecule. We look and we see that the dimensions are something that match the lipid bilayer, right? So what I say here is that rules are made to be broken. That is to say, we usually bury our hydrophobic surface area away from the solvent, and here it looks like we're exposing hydrophobic surface area, but it's only sort of a breakage of the rules because we're still burying that nonpolar surface area, but now it's inside the membrane itself. And it takes about 20 residues of alpha helix to traverse the membrane. Harkening back to our lecture on primary structure of proteins, here's a hydropathy plot again, and this having nothing to do with the three-dimensional structure, only looking at the amino acids and ranking those amino acids based on their hydropathy, and what we see is a very clear pattern. Nonpolar, polar, nonpolar, polar, nonpolar, polar. So we can identify the regions of the molecule that are clearly nonpolar, and then we can see the polar ends of the alpha helices. And the key here being that we can use amino acid sequence itself to predict what kind of molecule might actually be an integral membrane or membrane spanning protein. And clearly here we see a pattern that indicates that this molecule would be a membrane spanning protein. So another interesting idea is that proteins are composed of domains. And what is a domain? Well, it's a sort of a structural subunit that structural subunits are then assembled into the larger molecule. This is the C-Sarc tyrosine kinase. There are 3 distinct subunits. You can see them, but I'll point them out for you. This is a molecule that's involved in various carcinomas. It's composed of a tyrosine kinase. Now, this is a bit of more terminology. A kinase is a molecule that adds phosphoryl groups to proteins. In this case, adds a phosphate to the hydroxyl of tyrosines. But in addition to the tyrosine kinase, the enzyme portion of the molecule, there is an SH2 domain and an SH3 domain, and both of these are protein-protein interaction domains. So 3 distinct structural subunits. What this allows us to do is take our tyrosine kinase out of its context, place it in a different context depending on what kind of SH2 and SH3 domains we have, and suddenly we have assembled a new molecule that might actually phosphorylate a different target molecule. So what we see is modular construction. We have a relatively simple toolbox that we can assemble in different ways through domain swapping or gene duplication to give rise to new structures and new functions. We will see plenty of examples of this kind of modular construction as we proceed. Now, this lecture is ostensibly about tertiary structure, but I violated that at the very beginning by introducing a protein, GCN4, that was an example of quaternary structure. And in quaternary structure, this is an assemblage of three-dimensional molecules, three-dimensional structures. And in this case, the quaternary structure doesn't exist in the absence of one of those. So we don't fold in the absence of the other molecule in this case. And here again is a leucine zipper protein that interacts with a DNA molecule. And here is an interesting paper if you want to go read about it. I might mention it a little bit in class and we could discuss it. The details of the paper are not so important, but I wanted to pull some figures from that paper to help you understand how this quaternary structure is assembled. Very intimately constructed, involving both hydrophobic packing through the leucine zipper, the hydrophobic interdigitation that I mentioned earlier, and through salt bridges. Now, what's a salt bridge? A salt bridge is an ionic interaction between oppositely charged species, in this case an acid and a base. Harkening back to our understanding of the ionization state of amino acid side chains under physiological conditions. What do we have? We have an arginine, positively charged, interacting with a glutamic acid, negatively charged. So we have a series of salt bridges that are very stable, highly stabilizing, and linking across the strands. So the structure in this case is held together in a quaternary fashion by interactions that cross from one polypeptide chain to another. So that's the salt bridge part of it here, and you can see here's a lysine and a glutamic acid here, and then a glutamic acid lysine interaction here. So overall, six salt bridges holding these two helices together. But that's not all. There's also the leucine zipper. And here's the sequence again. I've taken away the salt bridges and I've highlighted the leucines, and we see that every 7 residues or so in this region of the molecule, there are leucines that form intimate hydrophobic interactions, again, across strands. So highly stabilizing interactions. If you want to read more about the details of that structure, you can look here. Again, we're taking it completely out of context. We don't care, in this case, what the DNA binding is all about or what's happening when it binds DNA. We're just looking at how we assemble that quaternary structure. But we can look more closely at protein-DNA interactions if we want. They're certainly very interesting. Taking, again, a couple of figures straight out of this paper, and I'll post this paper again for you to read if you're interested. Moving away from the leucine zipperin to the details of the protein DNA interactions. Now, let me point out a few interesting details here. You see the interaction of this helix with the major groove of DNA. I mentioned this in one of the earlier lectures. One of the beautiful things about this major groove, minor groove structure of the DNA double helix is that sometimes we can accommodate an alpha helix perfectly into that major groove, and there are other cases where we bind it into the minor groove. But in any case, let's look at the details of what's stabilizing that interaction, and we're going to look at specific interactions now, remembering that there are also non-specific interactions that stabilize this protein. So here's the structure of the DNA that's recognized by the protein, and we see when we look at the crystal structure that this amino acid interacts directly with this cytosine through a specific hydrogen bond. Right? We can see that the DNA is donating the hydrogen bond to the carbonyl, and here we see that same amino acid interacting again, forming another hydrogen bond, and that's across to the other strand of the DNA, to this thymine. So, oops, no, come back. So to clarify, these amidated acid side chains are really interesting because it allows us to both accept and donate a hydrogen bond, and this can be a very specific interaction with the DNA, providing for the sequence specificity of the protein. Okay, that brings us to the end of this discussion of protein tertiary and quaternary interactions. We'll finish up this series of lectures with a discussion of protein purification. Thanks. See you there.